Nice train wreck to start off with. Does that look okay for everybody? Okay, cool. I see some thumbs up. So today I'm going to be talking about a skill that I've been developing over the past few months during my time in the Joseph's lab, and I'd really like feedback from all of you on maybe better ways to communicate this type of analysis I'll be talking about, just K-mer-based GWAS, or if you see ways I could improve it at all, that would, I'd also welcome any feedback like that, because this analysis will be in some projects for my dissertation. So really, I'd appreciate any kind of feedback you have. I have this GitHub link, which, let's see, should have copied it so I can drop it in the chat here. I've posted like all the scripts I've written to do this analysis on GitHub. I tried to document them pretty well, but the idea is that if you're interested in doing this type of analysis, maybe some of that code could help save you a little bit of time. But of course, if you ever want to do something like this, please feel free to reach out to me. I'd be more than happy to talk with you about it or work with you. I really enjoy doing this sort of thing. Okay, so I also thought this could be useful for some of you, because I know either a lot of us have done GWAS in the past or are proposing to do it some more with some of our upcoming projects. So hopefully this gives you some interesting ideas for other things you can do with GWAS. Okay, so if I can advance here. If you've sat in on a presentation of mine before, you've probably had to hear me talk about GWAS. I mean, K-mer-based GWAS specifically. Hopefully I'm not annoying you too much yet, but I'm gonna briefly talk about what it is again so that everyone's on the same page. So a lot of us are probably familiar with what more traditional SNP-based approaches to GWAS are, where you correlate the presence-absence patterns of different SNP alleles with your phenotype of interest to point at different regions of a genome that might be implicated in whatever trait you're studying. So like, for example, if you were interested in the genetic basis of this purple color trait, and you found that all purple individuals had an A at this specific position in their genome, that could suggest that either this adenine or the sequence surrounding it influences that purple phenotype in some way. And sort of the main data structure you work with when you do this type of analysis is a SNP matrix like this, where every column gives the genotypes for different individuals, and each row represents the different SNP. And then there's lots of ones and zeros. The ones show whether an individual has the alternative allele for that specific SNP, and the zeros indicate whether the individual has a reference allele for that specific SNP. And we can do a slight variation on this approach that utilizes the same kinds of models as SNP-based GWAS, but can give us some extra insights. And that variant is K-mer-based GWAS. So a K-mer is just a subsequence of a read. So if you gather DNA sequence in chunks called reads, and you look at just a small string of letters within a single read, that's called a K-mer. So a subsequence that's 9 base pairs long would be a 9-mer. An 8 base pair long subsequence would be an 8-mer, and so on. So instead of correlating the presence-absence patterns of these SNPs with your phenotype of interest, you instead correlate the presence-absence patterns of these K-mers. So for example, for our purple trait, this purple individual is the only one that has this specific K-mer in their genome. So that could be some evidence that this K-mer somehow underlies whatever, or underlies this purple trait. So in this case, the data structure we have is slightly different. The columns still represent different individuals, but now the rows are K-mers. And the ones and zeros indicate whether a particular K-mer is present or absent from an individual's genome. So the AATTGC K-mer, for example, that is present in the genomes of all 3 individuals here. So each individual gets a 1, but some of the K-mers have zeros for individuals where that particular K-mer is absent. So really these 2 types of data structures are basically the same. They're just matrices of 1s and 0s, and you can apply the same types of association models for both of them. It's just that the type of insights you gain from one could be different from the other. So that's kind of what K-mer-based GWAS is in a nutshell, but I just told you about how similar the 2 are, so why would you want to consider doing one in the first place? And really there are lots of benefits to doing a K-mer-based GWAS as opposed to a more traditional approach. Kind of the primary benefit is that these K-mers capture the influences of genetic variants on your phenotype besides just SNPs. So there are lots of other types of variants out there. There are presence-absence variants, structural variants, and so on, all of which can influence quantitative phenotypes or pretty much any phenotype you might be interested in studying. These types of variants have been implicated in. And by looking beyond just SNPs, a K-mer-based GWAS ends up being much more powerful than a SNP-based GWAS in many instances. And that's because, again, we're capturing different additional types of genetic variants and their influences on the phenotypes we're studying. We're not just limited to SNPs anymore. A second big benefit of K-mer-based GWAS is that it doesn't need a reference genome in order to run. So a lot of us are working on Arabidopsis or species that have pretty nice reference genomes already. But if you go in a direction where you start studying more rare plant or microbe species that don't have reference genomes, the more traditional SNP-based analysis is off limits to you because that requires a reference genome in order to identify SNPs. So a K-mer-based GWAS could be an alternative approach that you could use in those instances where you don't have a reference genome. In the case that you do have a reference genome, though, a K-mer-based GWAS is still a useful thing to apply because again, the analysis can still be much more powerful than a SNP-based approach, and the reference genome that you have can help with interpretation of your results. And then finally, a K-mer-based GWAS uses pretty much the same starting data as a SNP-based GWAS. It's just whole genome sequencing data. So if you're already going to be doing a SNP-based GWAS, it could be relatively easy to just tack on a K-mer-based GWAS to that as well. It wouldn't require a whole lot of extra work. In terms of, you wouldn't have to acquire any additional sources of data. You just have to do some slightly different analyses. But there are also some big drawbacks to this kind of approach. I usually just talk about how great this approach is, but it has some caveats too. The primary caveat I'd say is that a K-mer-based GWAS involves a really high multiple testing burden. So in a usual SNP-based approach, you calculate associations between maybe a few million K-mers, between maybe a few million SNPs and your phenotype of interest. But in a K-mer-based GWAS, you'll be dealing with hundreds of millions of K-mers and calculating hundreds of millions of associations. So there's a really high probability that you'll get lots of false positive results simply because of the number of tests you're running. And you have to account for that, which I'll talk a little bit more about later. And then finally, the biggest drawback to a K-mer-based approach is that the results of a K-mer-based approach are much more difficult to interpret than a more traditional SNP-based approach. With a SNP-based GWAS, it's relatively easy to use like automatic annotation software to figure out why some of your SNPs are correlated with your phenotype of interest. Are the SNPs in a gene somewhere? Do they cause a missense mutation or maybe a nonsense mutation or things like that. But in a k-mer-based GWAS, usually you don't have anything like that. And instead, you're stuck with looking at alignments between sequences to try to interpret why a specific k-mer is associated with your phenotype of interest, which can be challenging. So that's k-mer-based GWAS in a nutshell, and I've kind of broken up the main steps of the analysis into 5 parts. Again, all the scripts to do each of the parts is on GitHub. But does anyone have questions on like k-mer-based GWAS or what it is before I keep going? Yeah, Maya, go ahead.
Sorry, if you are doing a k-mer-based GWAS, would you assume that you're also capturing SNPs that would show up in like a SNP-based GWAS? So you're just like layering on.
Yeah. Okay. Yeah, absolutely. Absolutely. So yeah, these k-mers will capture the influence of all these types of genetic variants in addition to SNPs. Yeah. And in that way, there can often be lots of overlap between a SNP-based GWAS and a k-mer-based GWAS. But they will also have some differences. Does that make sense? Cool. All right. So the first part of doing this analysis is pretty similar to what you would do if you were going to do a more traditional SNP-based approach. And that's simply getting lots of sequencing data and doing some quality checks on it and filtering out the bad sequencing data versus the better sequencing data. And that's what the first 6 scripts on the GitHub do. And a lot of them use a pretty simple trick to try to process lots of sequencing data really quickly. And they do this by trying to split some of the simple operations you'll be doing over multiple cores. So the main input to the scripts I provided is just a list of FASTQ file names. So a list of sequencing data files. This example here, I just picked some random FASTQ file names from the short read archive. But you could have a different list of FASTQ file names as well. And the first thing is that this big list gets split up into smaller sublists. And then each list is looped over on a different core where each FASTQ file is manipulated inside that loop. So on different cores, you might have a loop going over this list and downloading each of these files, then running some quality checks and some adapter trimming techniques with cutadapt. And you would have that split up over many cores because you would like the analysis to finish before you graduate. And so a lot of future steps in the analysis use this same trick to make things go pretty quickly. But I just wanted to mention that here. So yeah, this trick is used really frequently throughout the rest of the scripts I provided to speed things up quite a lot. So that's the first main takeaway for doing this kind of analysis, is that you want to parallelize every step you possibly can, because in comparison to a SNP-based approach, especially for some of the later steps, it can require some additional computation that you wouldn't usually encounter with a SNP-based approach. But the next part is actually creating this data structure that we're going to be dealing with in our actual GWAS analysis, the presence-absence matrix. And this is what scripts 7 and 8 do. So now we're going to actually be working with k-mers, and you have to start by counting the frequencies of different k-mers in your whole genome sequencing data set. There are really 2 different tools you can use for this, KMC and Jellyfish. They're both pretty much identical in terms of performance. They're both really fast. They're both great. KMC has some additional functions that are really useful for later steps in the analysis, so that's what I recommend. but I'm sure there are other programs out there you can use as well. The way that these programs work is that they take each read of sequence data. So here I've just listed out a random string of letters as a read, and it takes a sliding window, and it starts at the beginning of the read, looks at the k-mer within the window, and adds 1 to that k-mer's frequency in a growing list of k-mers. That's a growing list that's keeping track of all the frequencies of different k-mers in your data set. So when this window starts here, it finds this ATGC k-mer, and it adds 1 to that k-mer's frequency in a huge list. So then this window would shift over 1 base pair. I didn't animate it, but this window shifts over 1 base pair, and then the frequency of that k-mer gets counted, and then another 1 base pair shift, and so on. And you just repeat that until you go through all the sequencing data. But this really highlights the first big decision you have to make in this analysis, and that is deciding how long the k-mers that you're counting are going to be, or in other words, how wide this window is here. So here I have a window that's 8 base pairs long. In reality, you want a much longer k-mer, but you have to decide, all right, how long is that k-mer going to be? And really, the answer is that you want this k-mer length to be as long as the length of your shortest reads. So what I mean by that is, if, for example, the shortest reads in your data set are 31 base pairs, then the length of k-mers you want to count is 31 base pairs. Because k-mers are by definition subsequences within a read, it doesn't make much sense to try to count k-mers that are longer than some of your reads. And in most papers I've read, they look at k-mers that are 30 to 40 base pairs long. So that's what I recommend. I use 31 if I'm dealing with Arabidopsis data, because that works pretty well. So if you're trying things out, I'd start in that range. And then the next big decision you have to make is at what frequency should a k-mer be considered present in your data set? So if going back to this example up here, this ATGC k-mer, so far the program has seen this k-mer occur once in our data set because it found it in this window. But it could be easy for that one instance to just be generated by like a sequencing error. So usually you want a k-mer to be identified multiple times in your sequencing data set before you consider it present. So as many papers I've read use a frequency threshold of 10. So if a k-mer occurs 10 times in a sequencing data set, then it's considered present, and it has a 1 assigned to it in the k-mer presence absence matrix. Does that kind of make sense to people? Yeah, Nate, go ahead.
What kind of sequencing projects create, let me rephrase this question. What kind of sequencing reads do you use? Do you use like single end? Do you use paired end? Do you use like RADseq? What kind of projects?
Yeah, that's a great question. You can use single end or paired end. Often, if you're working with Arabidopsis data, you'll probably be working with a mixture of paired end and single end data. So really, you can use any type of sequencing data. I can't think of a reason for why you would not want to use a certain type of sequencing. A k-mer-based GWAS can be really useful in instances where you enrich for particular types of sequences in your data set. Like if you are interested in like specific kinds of disease resistance genes in plants that have really similar sequences. You can use more targeted kinds of sequencing data still with a k-mer-based GWAS. That's been done before. Yeah, I can't think of a reason for omitting one type of sequencing.
Just a very short follow-up. For paired end, are these k-mers connected in any way, or are they effectively split so they're single end?
Well, I'd say that's I'd say that well, I'd say that's really what happens is that for paired AND data, if a k-mer occurs a total of 10 times looking at both the forward and reverse reads, so say you find the k-mer 5 times in the forward reads and 5 times in the reverse reads, that would be enough to consider that k-mer present, because it occurs 10 times in total in that sequencing data set. Does that make sense? Is that okay? Is that your question? I think. Okay, cool. So yeah, Sophie, go ahead. Oh, they left. Never mind. Okay, so okay. So next step is to actually make this matrix. So we've counted up all these different k-mers. And now we have to actually make the presence absence matrix. So currently what you'll probably have looks something like this for each set of sequencing data. So this is one set of FASTQ files. This is another set of FASTQ files. And each set will have a list of k-mers, a list of k-mers that were found to be present in that data set. So yeah, what you want to do is take all these different lists and merge them together into a big matrix. There'll be lots of these files, so it'd be impossible to do this merge in R. Really what you want to use is bash because it's much faster. And so these scripts I have here, 9.0 and 9.1, they'll use a bash join function to split up this merging process over multiple cores and multiple stages to make that merge process go really fast. So that's the second main takeaway, is that you don't want to try to handle any of this kind of data in R or Python because it'd be really slow. Any questions on that? Doesn't look like it. Okay, so we're not at doing the actual GWAS yet, because a lot of the k-mers that we've counted, at this point you've probably counted, hundreds of millions of k-mers, maybe billions, and a lot of those k-mers aren't actually going to be really useful for any type of association analysis. So the first thing we're going to do is apply some additional filtering steps. So say we have this matrix here where, again, we have these k-mers, and then the ones and zeros give their presence absence patterns. A lot of k-mers, for example, will have identical presence absence patterns across a lot of individuals. So these two k-mers here, for example, they were present in all of these sequenced individuals. So they have the same presence absence patterns, and calculating correlations for both of them would be kind of redundant. So instead, the solution is to just drop one of them. So in this case, you might just drop this k-mer right here and keep this AAA k-mer. Yeah, a lot of the k-mers you work with or that you count will have that problem of either being present in a lot of individuals or only present in one individual. Like these two k-mers, they were only found in this one individual. And so in those cases, it's useful to drop k-mers that have those redundant presence absence patterns. Yeah, go ahead, Maya.
Are you using a specific threshold for what is an identical presence absence pattern? Is it every individual, or every individual minus one?
Yeah, that's a good question. So what I do is just it's an exact match threshold. So if this k-mer and this k-mer have identical presence absence patterns across all the individuals in the matrix, then that is the instance where you would just drop one of them and keep the other. Does that make sense? So they have to match it all for all of the individuals. They have to have identical presence absence patterns. Yeah, there's not yeah, hopefully that makes sense. Okay. So the next filter is similar to like a minor allele frequency threshold, where you're going to get rid of k-mers that are either really rare or really common in your data set. And that's because they may not give super reliable correlations, or maybe those rare k-mers are just due to sequencing errors. That's possible. And usually what I do is remove k-mers that are either present in fewer than 5% of the individuals or more than 95% of individuals. So it's kind of analogous to like a minor allele frequency threshold of 0.05 that you might apply in a usual SNP-based approach. And then you'll convert everything into the proper format. And finally, hopefully you'll just be left with a few 100 million k-mers instead of billions. Progress is, I guess that's what that is. So this is scripts 10 to 13. Okay, so filter out the majority of k-mers that you count, because a lot of them won't be useful. You'll still be left with hundreds of millions probably. And this is the point where you will finally do the GWAS. Or at least we're getting close to that. So first you have to deal with the issue of kinship matrix estimation. So this is a matrix that accounts for relatedness among the individuals in your diversity panel when calculating correlations between k-mers and your phenotypes. You don't want to use all of the k-mers that you have to estimate this matrix, because that will require a ton of memory, and whatever estimation algorithm you're using will probably crap out on you. So instead, you want to use some kind of subset of the k-mers that you have in your final filtered data set. I usually use a subset of 10 million k-mers. That seems to work pretty well in Arabidopsis, and I chose 10 million kind of because that's sort of the upper limit of most SNP-based GWAS analyses in plants. They have, just a few million k-mers, a few million SNPs often. And now finally, after all this work, you can actually do the GWAS. And a lot of GWAS models that you may come across will be looking for variants that are biallelic. So SNPs can kind of have two states. They can either be heterozygous in an individual, or they can be homozygous. K-mers don't have that same property. It doesn't really mean anything for an individual to be homozygous for a particular k-mer, because you don't have that information. And so really you want to try to overcome that inbuilt tendency in GWAS models that look for that try to determine whether a variant is homozygous or heterozygous. And you can do that by using this not SNP option in GEMMA, for example. Once you fit the model, you'll probably get many different significant k-mers, probably in the realm of a few thousand, which can be quite a bit to interpret. But then finally, you can visualize the results. You can use some more standard ways to visualize GWAS plots, like Manhattan plots and QQ plots, although you'll probably only want to plot a subset of the k-mers that you have, because again, you'll be dealing with a lot and you don't want it to make a massive image file. This is kind of an example of how I've been visualizing k-mer-based GWAS. It's probably not the best way to do it. On the left here, this is a Manhattan plot from a more typical SNP-based GWAS, where each point represents a different SNP, and it has each SNP has a location on a genome. And then the height of the point shows the significance of the SNP's correlation with the phenotype. On the right here, this is a similar type of plot, but now on the horizontal axis, I have different k-mers ordered alphabetically. So you can kind of see like k-mers that occur at certain positions of the alphabet where they where they fall in terms of significance. It's kind of like, I'm not sure how terribly informative that is, but it's one thing you can do if a person, maybe if a reviewer demands to see some kind of Manhattan plot, if you're doing a GWAS, which can happen. So does that kind of make sense to people so far? I don't know if I'm going fast or slow, but okay. Well, I don't see anyone.
Can I have another question?
Yeah, go for it, of
This
course.
might be like really obvious, but why can't you plot the k-mers by location like you do with the SNP-based GWAS?
Yeah, that's a good point. In the case where you don't have a reference genome, which could be a situation, a k-mer-based GWAS doesn't require a reference genome. You might not know where a k-mer falls in the genome simply because you don't have that reference. If you do have a reference genome, you could do something like you could try to align all of the k-mers in your data set to places in the reference genome to see where they fall in the reference genome, and maybe base your plotting that way by where your k-mers align to. But again, there will be k-mers that aren't present in your reference genome. So there can be, like, say, variants that are present in some accessions but are absent in the reference. Those variants that aren't found in the reference could still be important for your phenotype of interest, but they won't have any basis in your reference genome, if that kind of makes sense. So you could kind of plot the k-mers based on location, but it kind of gets away from one of the tenets of the analysis, which is not trying to base everything on a reference genome, if that kind of makes sense. All right, cool. Okay, now finally, we can actually try to interpret the results, which is probably the hardest part of this entire process. And really, it's where k-mer-based GWAS kind of falls short relative to SNP-based GWAS, is that it's harder to interpret the results. So yeah, up to this point, we may have thought that we're done, but we still have two massive problems trying to interpret the results, as I've said. But also, more likely than not, you'll have many thousands upon thousands of significant k-mers that are associated with your phenotype of interest. And it could be really hard to try to interpret every last one of those, especially if you are impatient like me. So we want to figure out a way to narrow down the set of k-mers that we're going to try to interpret, and try to develop just like a high confidence set that we're really certain are associated with our phenotype of interest. And there are a few different things you can do to accomplish this. First, you could apply a really strict multiple testing threshold. That's probably what you're going to end up doing to begin with. But in addition, there are some other things I think you should try. Even after applying really strict multiple testing thresholds, you'll probably still have lots of k-mers associated with your trait of interest. Another thing you could try is applying multiple types of GWAS models to the same data set. So, sorry, I just dropped my mouse. But for example, you might, one thing I often do is apply like a univariate GWAS model, a multivariate model, and some other types of models all on the same data set or a similar data set, and look at just the intersection of sets of significant k-mers from all those models to try to identify, again, just k-mers that we are really, really certain are strongly correlated with our phenotypes of interest. Some papers will do a weird thing. We'll go through multiple rounds of GWAS. Like we'll do one round of GWAS to identify k-mers that are pretty strongly associated with their trait of interest. Then they'll take just those strongly associated k-mers and do a second round of GWAS on just those. I don't know, I feel like that's kind of weird, but some papers do that. And it's something you could consider. Another thing that I would recommend is including covariates. in your GWAS model. One important covariate to probably include in most models is sequencing coverage. So a k-mer's frequency in a data set is largely determined by how much coverage there is in your sequencing data. And coverage usually varies between the individuals in your data set. So you want to make sure that the associations you're getting aren't simply due to differences in coverage between individuals. So it's good to include sequencing coverage as a covariate in your model, and that will decrease the number of significant k-mers you get. Okay, and I think at the last stretch here, so the last 3 scripts are all about trying to interpret the results of k-mer-based GWAS. And really, your interpretation is the way you go about interpretation is going to be a little different depending on whether you have a reference genome or not. So if you have a reference genome, you have really 3 different options. The first is that you can try to map the significant k-mers themselves back to your reference genome to see where they fall. Problem with that is that the k-mers you're using could be, excuse me, they'll likely be too short to align super well to a unique location in the genome. So it could be hard to interpret those. One thing you could try instead is mapping whole genome sequencing reads containing significant k-mers back to your reference genome. So the reads will be slightly longer than the k-mers and you can get some better alignments. But again, the problem you might face there is reads not aligning to unique locations in a genome. These are both methods I've seen other papers use, so that's why I'm listing them here. And then finally, this is what I usually do. Instead, what I do is I take whole genome sequencing reads that contain the significant k-mers and assemble those reads into big scaffolds using an assembly program like Spades. And then you can take that really long scaffold and align that to the reference genome. And usually those scaffolds will align to just 1 or 2 places in the genome because they'll be fairly long, and it'll be a little easier to interpret those results. One thing you'll have to think about though is trying to figure out or trying to filter out scaffolds that were generated from assembly errors. But that's the approach I recommend. All right, if you don't have a reference genome, interpretation is a lot more difficult. So I said a k-mer-based GWAS analysis doesn't require a reference genome, but if you have one, it's a huge benefit because instead what you're left with is what I think you'll probably just end up doing, like I listed up here, assembling whole genome sequencing reads with your significant k-mers into scaffolds, and then maybe just doing some sort of blast search to find some previously known sequences that are similar to your scaffolds to try to interpret them that way. There might be better ways of doing that. I can't really think of any, but it could be really difficult to try to interpret the results without a reference genome, although it is possible. So yeah, at the end of the day, you'll end up looking at alignments between these scaffolds and either a reference genome or some other database of sequences to try to figure out what the different k-mers are representing and why they're coming up as significantly associated with your trait. Okay, and that's everything. So I guess the kind of main takeaway from that last part is to try to narrow down the number of k-mers that you're looking at, and try to combine multiple types of approaches to GWAS. So one thing I like to do is combine k-mer-based GWAS with a SNP-based GWAS, and do things that way. Because although k-mer-based GWAS is better in some ways than SNP-based, they can be kind of complementary. So think about using multiple approaches instead of just this one approach, because I told you it's better. Okay, so this last file, this is just a summary of the different kind of software you might need to run the analysis. We don't have to look at that. these are some useful references. I'm going to put these slides up on GitHub later so that you can reference them later if you want. These are some papers I found really helpful for doing K-mer-based GWAS. There aren't many papers that do this type of analysis in plants, at least that I can find, but here are some helpful ones. There are also a lot of papers in microbes that try to do K-mer-based GWAS, and those are really interesting, so you can check some of those out as well. Okay, and I think that's everything. So thanks for sticking with me.
Thanks, and I will be using your code.
Cool, yeah. If you ever have like any problems running it, or I don't know, anything at all, if you want to do this, please talk to me, and I'd be more than happy to offer pointers or work with you or anything like that. So
Miles, I had a devil's advocate kind of question.
go for it.
So you said that your approach at the very end to try to find these actual associations is to assemble scaffolds with with the reads that the K-mers came from. How many reads do you find it to be useful to make these scaffolds? Because I can, and I'm kind of like leading into to what I think is the answer. Like I can feel like if there's you can find cases where there's like very, what am I trying to say? Like a K-mer that's very associated with whatever trait you're interested in, and it's like a loner read. You're not going to be able to assemble that, right? And so how would you get around that?
Yeah, absolutely. So that's a great question. So usually the K-mers that you include in this analysis, they will have reached some kind of frequency threshold in your data set. So kind of going back to the beginning, usually these K-mers will have occurred at least 10 times in your whole genome sequencing data set. And in my experience working with Arabidopsis data, I usually end up, for a particular K-mer, I get somewhere in the realm of like tens of thousands of reads that contain that K-mer, and do assemblies with that. Sometimes on the order of like 100,000 or so. And I'm able to get scaffolds that are a few hundred base pairs to maybe 1,000 base pairs long at their longest, and get usually unique alignments. Did that answer your question, or did I dodge it?
Nope. Oh, that was good.
Okay. All right, well, I can go ahead and stop sharing, but thanks everybody for listening.
Yeah, thank you for that, Miles. That was awesome. I'm going to stop recording if you don't mind.
Yeah, that sounds
Is that
great.
cool?